plaque is an area on a plate where no live bacterial cells are growing. These are some examples of our phage plaques. These plaques are formed from a single phage infecting a bacterial cell, replicating and lysing the bacteria. The phage then move to surrounding bacterial cells and lyse them, forming a plaque.
So we use top agar when we work with our bacteriophage because top agar allows us to mix the bacteria and the phage together and then spread it out on a plate and allow us to seize the results. What we usually do is we'll make top agar that's twice the concentration we typically use it at and allow it to solidify at room temperature for storage. Not only does this store well, but when we are ready to use it, we can melt it in the microwave and then it becomes liquid, but it's too hot to use. But since it's twice the concentration, we'll mix it 1-to-1 with typical liquid broth media, and we'll do it in a 50 milliliter test tube that's sterile, or you can use a sterile flask or beaker or jar. And then the top agar will be closer to room temperature and better to use. It won't kill your bacteria. So here we're mixing our top agar so that this really warm 2x top agar can be diluted down to 1x and become closer to room temperature and easier to use. Saves a lot of time so we don't have to wait for it to cool down. So we'll mix it up. You can check if it's ready to use, if you can hold it without it being too warm to hold. It's about good. Okay, so now we'll take our bacterial sample that's been infecting the phage, and we'll grab a 5 milliliter pipette, and we will remove 4.5 milliliters of top agar. It's been mixed and is now cool. And we'll put it in our bacteria and phage. It's always good to mix a couple times, just so you make sure the bacteria is spread out in the mixture. And then we'll put it on our agar plate. I usually like to cover up the plate with the lid just so you avoid contamination. So you put the agar in. Notice we got a bubble here. We'll need to suck that up because bubbles can look like plaques, and that can make your results difficult to analyze. That's good. One of the other things that you can use top agar for is varying the concentration of the agar in the broth to change the size of plaques. This is useful if you're selecting for different plaque sizes. This is an example of T4 phage where we use different top agar inside different sizes of plaques. This is 0.15%, and the plaques were pretty small, and they get bigger as you move up to 0.1, and bigger still when you move to 0.075%. The same principle can be applied when trying to find small phage, because you can increase the percentage of agar and the plaques get really, really small.
To isolate a single phage from a plate full of plaques, we put a ml of broth into an Eppendorf tube. And then we find a plaque that we like. For example, we have this little teeny one, which is more likely a larger phage because it hasn't moved around very much to make a very big plaque. And we'll take the end of an Eppendorf, not an Eppendorf, end of a pipette tip, and we will ever so carefully stick it right in where the plaque is. And put it right into the broth and swirl it around so all the phage can get out in there. And then with this, it's dilute enough to where we can take about 10 microliters from here and infect directly into the half mil of bacteria. To test whether or not we have phage in our sample, we'll do what's called a spot test. So we first draw a grid on the back of our plate, and we'll take some of the sample that we think has phage in it. We'll take just about 4 or 5 microliters, and we'll put it onto this plate that has only bacteria in it, no phage. And we'll just put a tiny little dot on there. And then we wait for it to sink in, because if you tip it over, then it will all go running, and it's not quite as pretty. So you just do this with every one of your different kinds of what you think has phage in it, and then if a plaque forms at the end, then you have phage. This is an example of a good spot test. Each one of our samples did actually have phage in it, so you can see the plaques that they formed. In these two, we don't have anything, so no phage. Over here we have a plate that we tipped over too soon. So the spots didn't sink into the agar, and so when it got tipped over, they all cried, sort of thing. So not quite as pretty, but there is phage in there, since there's lysis.